Yeah, today I'm going to talk to you about the role of natural history biorepository in host pathogen studies and documenting the Auchenorrhyncha Phytoplasma association through biorepository and how to use them for disease risk assessment using machine learning approach with preliminary data that we obtained for our project. The next step. So, first of all, according to the National Cancer Institute, a biorepository is a facility that collects, catalogs, and stores samples of biological material of any origin, animals, human, plants, for laboratory research. Specifically, museum biorepository preserve material for projects in several research fields, such as biodiversity, phylogenetics, population genetics, toxicology, environmental monitoring, and pathology. And also, we know that biorepository play an important role in tracking host pathogen association. For the host pathogen association point of view, catalogs, records, and voucher specimens are the primary source for tracking past association and infer evolutionary pathways, documenting association in no longer accessible collection sites, revising the taxonomy status of the associates, which is really important, resampling the association over time when new technology became available. So when documenting host pathogen association, preserving voucher specimens is a critical step. The practice of vouchering, as you know, is performed mainly by taxonomy that are interested in morphological studies and is often overlooked by other subdisciplines such as genomics and epidemiology. And here in this figure, I'm reporting a very interesting paper where they basically mapped how many genome deposited. I have a voucher reference associated with it, and actually, so sadly, just 11% of them have a voucher. So, as pointed out by Dan Brooks in 1994, parasitologists should deposit voucher specimens of all species examined in the course of their survey, inventory studies, including purely ecological field studies, but unfortunately, this rarely happens. Here I reported a couple of examples from the United States, and on the left we have the Aaron Mantel Laboratory of Parasitology at the University of Nebraska. The museum is preserving voucher of pathogen associated with mammals mainly, several kind of elements, protozoa, and they have over 90,000 collection objects and from all over the world. And then on the right, of course, we have Illinois Natural History Survey insect collection, and we recently started to focus on the host parasites association. So let's have a look briefly to the collection in Natural History Survey. So we have over 1 million collection objects, among them Coleoptera, Collembola, Diptera, Emiptera, Hymenoptera, Lepidoptera, Procoptera, Socophora, Trichoptera, and Tithanoptera, but of course we focus on Emiptera, and they are mainly preserved as dry specimens or in ethanol. And as feeders, they may harbor vector-borne pathogen, even if they are preserved in the collection. And this is why we recently started to document the presence of phytoplasmas in Auchenorrhyncha insects at the Illinois Natural History Survey. So in 2021, we published the first paper where basically we discovered the presence of phytoplasma in leafhopper preserved in museum biorepository. So after that, immediately after, we applied for an NSF project, and we recently started, and we basically are revealing and discover diversity of phytoplasmas, but our point is to screen insects, and specifically insects that are preserved in biorepository. The main goal is to discover the true biodiversity of phytoplasma that we hypothesized to be underestimated, and then assess potential biohazard. So today I'm going to present preliminary results of risk assessment, just because we recently started the project. So we have a few data. We don't have a characterization of phytoplasma yet, as well characterization of vector, insect vector. So anyway, at Illinois Laboratory Survey, we have, so in a literature lab, we have more than 1,000 bulk samples of emitted insects collected between 1998, more or less 2080 worldwide, and they are waiting to be butchered and screened for the phytoplasma presence. And here I map on this raster analysis. I don't know if you can see the red points across the map. Those are the collecting events, the habitats that where the Chris and their students collected these specimens. And as you can see here, the points are exactly on the non-crop land. So this is just to reveal that they are mainly collecting in natural areas. So now the point is, why we need to focus on natural areas, why it's so important for us to assess the disease and assess the outbreaks in natural areas. So actually, we have been working on this topic, and we provided evidence for crisis of emerging infection diseases stemming from the lack of comprehensive inventories and deep phylogenetic context. And also, more importantly, a narrow focus on single habitats. That, of course, in this case, is agroecosystem. But we also know that we live in a complex and hyper-connected ecosphere. So we recently published a paper where we formalized these concepts, and we are basically looking into each single habitats that harbor a diversity of potential pathogen, and more importantly in this area, which we call interfaces. And those areas are the most important area for the pathogen and the host to interact, to switch. In other subdisciplines, you may want to call that ecotone or border or whatever, it is the same, but the interfaces are really important. So some facts about natural areas and why they are so important for phytoplasma diseases. So we know that traditional studies in agroecosystem in general focus on plant phytoplasma association and only in symptomatic plants. And we also know that almost all known vectors are documented in agroecosystem or eventually surrounding ruderal habitats, and still vectors are known just for half of the known phytoplasma groups. And we also, after review of the literature, we discovered that only 10% of the previously documented association involved non-cultivated plants. So very few attempts have been made to document phytoplasma association in natural areas. For this reason, our project meant to cover this gap. So as I said, we are going in the future, in the next 2 years, to discover eventually through biodiversity of phytoplasma in natural areas, eventually to uncover new association with insects, emitted insects. But for today, I have just preliminary data, and I can run some risk assessment, which is also important and is part of our second goal. So this is the workflow of our project. So I'm going to focus on the first part for today, which basically involves, okay, you have a bunch of specimens on collection, you go through a selection process, then you go through a DNA extraction, and then from one side you go through the morphological study, the vouchering, and then the databasing thing, and then for the screening of phytoplasma from the DNA extract, you go through the routinely used molecular analysis, and then eventually detection, yes or no, and then eventually you go further. I'm not going to present today because we don't have data yet, but we are going to use ATE, which is anchored hybrid enrichment next gene approach that have been presented a lot during the past days from my colleague. So this is what we have. So this map represents the first sample of 2,000 specimens that have been screened till now. But not just that. This map also represents all the work that Chris and his student are doing in the last 30 years, which is amazing, covering more or less all over the world, and a lot of different natural habitats, and we have this great opportunity to actually summarize the data here. So I'm analyzing today 2,000 specimens, 1,062 collection events, and of course we screen insects individually, right? So here's some, summarizing the data, just to let you know what we have. So the collecting events are spread through 50 countries. We have 722 unique collecting events, and you see here 15, including more than 15 collecting events. And in here you have the distribution of the sampling efforts since 1995, and there are some years where the efforts was greater for some reason, from 2002 to 2009. And here, the distribution by method. So eventually we have still half of them that have to be assigned to a precise collecting method, but most of them were collected through malaise drop or sweetlets or light. And then we have a few fogging, plantra, preliters sample, just to mention. Of course, about the total analyzed, 71% are in the subfamily of Deltacephalaini, because lately Chris has been focusing a lot on this subfamily, so eventually this will be is a bias that we are aware of, and that will be fixed in the next 2 years, where we are screening other family, other orders, and subfamily, eventually including also Auchenorrhyncha. So more than 91% are male, and again, this is not surprising because morphological study, but we are going to fix that too, because we know that females may harbor a higher percentage of phytoplasmas. This is just the distribution across the tribes. So I may want to mention some: Patisanini, Deltacephalini, Scapuleini, Varalumini, Scaptopini, Opsini, belonging to the Deltacephalaini, and Erythroerini for the Tiflosibani, that are the most represented tribes. So, as I said, we screen for the presence of phytoplasma using the routine approach, qPCR. And we found that 13% of the insect samples are infected by phytoplasma, the black dots are those. And this is the distribution by country. We have some country that turns out to be more infected, like Israel, as you can see here, Portugal. But of course there are, for example, Mexico, Peru, that also have a good proportion of positive samples. And then we have the proportion of positive specimens by here, so nothing stay here. And then, importantly, the proportion by sex, which kind of give us the indication that eventually female may harbor much more phytoplasmas. This is the proportion of specimens by subfamily. Again, we have more Deltacephalaini until now, but we have some Piastaini also that may be surprising to analyze more. So for the risk assessment, we obviously have to choose a explanatory variable, so we need the variable to understand how environmental variable or other socioeconomical variable may or may not affect the presence of phytoplasmas in natural areas. So of course we got average annual temperature for watering. This is really important. We got the annual precipitation, and then we got altitude, and here basically with the raster analysis you retrieve the data from each of the black points, which are our collecting events, and you have the distribution of the variables, and you have your data basically. And then I retrieve data from the social economic data and application center. You can pretty much download every kind of data from this website. And what I was interested in here was the adjusted population density. And this is what we have. So of course, as you can see, the population density is pretty low overall because we are in a area. So this is what it is. There are a few outliers that we take care of, but this is what it is. And it varies from 0 to up to this red line, mainly is 5,000 number of person per square kilometer. This is an interesting index that I retrieved. It's the Environmental Performance Index. So globally we have a goal to reach, each country have to reach 60% of protected area. So they have been collecting data over time to see which country reach that goal. And I on purpose make the country confusing here so you don't match the country with the color. But I just want you to look at the general trend. There are countries here that has a very low percentage of this index here that vary from 0 to 100. The global goal is 16, and this doesn't change over time. We have other countries that start a little bit bad and then improve over time, and then we have countries that have this high standard. So go ahead. I also tried to retrieve the food insecurity data, but unfortunately we have just data for 3 countries in our data set. And then of course agricultural cropland, which is an index that varies from 0 to 1. And then land cover, which is also a nice index from the Millennium Ecosystem Assessment. And I got the land cover from 2000 and the projection 2050. Of course, as always, before running any kind of statistics, you have to correlate and see, eliminate eventually autocorrelated variables. So at the end, we kept population density, environmental quality index, cropland, precipitation, temperature, land cover in 2000 and 2050. So, why use a machine learning approach to study vector phytoplasma association? This is a good question. The machine learning has a lot of good properties. So for example, you will be able to analyze a large volume of data, the so-called big data, and as an increased processing power, like that. So we also got better results aimed at identifying pattern in data and predictive future scenario, which is also the purpose of the risk assessment, and then enable self-learning from the data, and then you apply that learning to new data sets without human intervention. Many people don't like that. And in the case of vector phytoplasma association in natural area, we can predict that we will be more likely that we will predict what will be the more likely set of insect sample that will be infected by phytoplasma, given a set of important predictors that are those that I showed you before. So overall, our objective is to use observed prevalence of phytoplasma infections in museum voucher that were collected in natural area worldwide to identify and associate the risk factor. So this is an overall overview of how machine learning works. I know that this technique has been mentioned by a student during the second day or first day, so I'm assuming that most of you knows how machine learning works. It's an artificial intelligence approach, so it's very nice because you can train the model, you can train the model with trained data, and then you can use new data to improve the performance of the model if you are not satisfied with the results. So, and also very good things as a power for prediction. So this is our data sets. We have response variable, which is infection, so negative, positive, and then we have predictors, that are those that I mentioned before. So statistically speaking, we have to check for the proportion of negative, positive insect sample, because this is called imbalance. So if you have a class imbalance ratio greater than 2, it's not good for the analysis. So you want to correct it. We actually have 6.5 in this data set, so, and likely we have several techniques to correct this problem, and we use one technique. I mean, I tested the various possibility, and it turns out that the best one is the oversampling method using the Rose algorithm. Machine learning gives you the possibility to compare different models, different algorithms, and this is usually what you do. And I compare 5 different models that are in different types with different parameters, but most importantly, 4 of them also tested with this adjustment for the imbalance data sets. So we have, for example, stochastic gradient boosting with and without the correction for the imbalance. We have random forest, that probably is something you are familiar with or without correction, and so on. So this is the results. What do you want to do? You want to run each model, and you want to verify which is the most important variable for the model. And as you can see here, I'm sorry for the acronym, but the highlighted variables are precipitation. On the top, there is always precipitation, and then there is population density, crop, blah, 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 and on the bottom as well. But what you would like to do, you would like to pick the predictors that are important for the majority of the models, because we know that they represent genuinely important predictors. So it turns out that are all but the environmental indicator index. So we retain precipitation, crop length, temperature, and population density. Okay? So, and then we want to test accuracy and precision of the model. So I'm not going into the detail here. I just highlighted the model that performed the best. This is random forest, RF, and Rose, which means random forest corrected for the imbalanced data sets, which are what we expected have to be done. This is a really great tool for machine learning. We call this confusion matrix. It's not meant to confuse you. So I'm going through the explanation of this matrix. So those are the predicted variables for the model, right? So you got negative. If the predicted are actually negative, you have true negative. If the model gives you negative, but actually they are positive, it's a false negative. If the model gives you positive, and they are actually positive, are true positive. But if they actually are negative, they are false positive. So this is a very powerful tool to let us know how well the model is doing in predicting. Because as I said, we model, we choose the most important variables that we predict. So again, it turns out that random forests perform pretty well with an imbalance correction as well. This is my last slide, and I hope I'm not out of time, because, and the, sorry, just fell behind decision tree. This is called decision tree, and this representation is pretty good for people that don't want to go beyond the detail of the model, but they want to have an overall explanation of the performance of the model. So every time that you have new data and you want to predict what's going on, you have to answer questions at each step, and this is pretty much what the decision tree do. So if you have a precipitation less than 200 and so, you go this way, and if you have cropland that is higher of this value, you go this way. If it's less of this value, you go this way. And if you have a temperature less than 90 degrees, you go this way, and you got a probability for that sample to be positive. So this is what it is, and this is how it works. Next step, and I'm done. So of course, we are screening more sample, and what we want to do from this bunch of steps below, which is the classification. So we are going to actually classify phytoplasma with next-gen, determine what vector have been ingesting internal plant. So we are going to reveal some useful information about insect plant association and so on. So we are planning to do it next in the next year. And then, of course, characterization, as I said, and then initial modeling approach, of course, because when you get the data from characterization, you have data from single species, because what I presented was just positive, negative, which is the overall, like the othering presented for the genus level approach, which is fine, what everyone do, but eventually, if you can go into the detail. And then I really would like to thank my student, which is an undergrad in neural engineering. She's actually doing a very important job, which is performing the same exact analysis in a database that I have been collecting for several years, exclusively on our ecosystem. So it will be really fun now to compare how these results are for natural areas versus our ecosystem, because as I said, we care about interfaces.